secondary structure prediction. We got to the point that simple methods as we have them had a problem of not reaching 100% accuracy, of getting beta wrong, beta at almost random percentages, and of having short segments. We improved all of that by using neural networks. They did not get closer to the 100%, but they did in fact improve our beta strand by sampling differently. And they got segments longer by sort of correlating, by sort of stacking up neural networks where the input of one is the output of one is the input to the next level, and sort of getting correlations and sort of seeing that residue on I and I plus, that helices have to have at least 4 residues. That's what the second level networks saw. But the main issue remained that still the performance was just about 60-something% in terms of Q3. Not much better. The question was how to put more information into the input. So in the SGSP curve, what you see, number of residues aligned, percentage sequence identity, 100 residues aligned. If more than 100 residues, more than 33 of the 100 residues are identical, in this realm that is blue, every pair of protein with more than 33 of their residues in 100 identical has similar structure. Now, let's turn that around. What do you believe happens if I take a protein of 100 residue and I just close my eyes and I say this, I pick 5 positions in the protein, and I will replace whatever amino acid I see at that position into another randomly chosen amino acid. Will that protein, so this is only 5, I say what we can change is 67. I change 5. Will that protein have the same structure?
I mean, it depends on the position of the 5 change.
Right. This is a very important point. That depends on what I change. Now, put it differently. Do you believe that if I just randomly take a position in a protein and I sort of randomly substituted that amino acid, and my statement is that has almost no effect on average? That's statement number 1. Statement number 2 is that it is in fact very unlikely that this variant has no effect, and that in fact it's more likely that the variant has an effect and will not survive. That is statement 2. Which one do you favor? Statement number 1 and statement 2. Most variants are neutral with respect to structure formation. Statement number 1. Statement number 2, most variants have, or many variants have an effect, and to have one that does not affect structure is very unlikely. Those are 2 extreme statements. You only have to pick from extremes. Reality is somewhere in between. But on which side is it more? Yeah? Anyone? Yeah?
I think so. My feeling tells me that I think it's more like the variant has no effect, because you must be quite unlucky to hit such a substitution with, so there's a variant that has a very bad influence.
So one way of saying that, so Thomas, what you say, if I mapped it to, there's a database, online mammalian inheritance in man, OMIM, that in fact collects examples for mutations that are really immediately, one mutation is leading to a disease of the carrier. Whoever has that variant has some particular disease. Most of these diseases are pretty serious. And when you look at them, there are many of those, but when you look at, we have 20,000 proteins, 20,000 times 400 amino acid, or position residues. So given that space, these are relatively few variants. So of all the possible variants that we could have, this is a tiny subset. Okay? So that sort of may indicate exactly, maybe support for what you say. So very few of the variants that we have are very bad. But actually we don't know that. Because we don't know that because they may not survive. Many of these may be so bad that we will never see them. We will not characterize them as OMIM variants because we now have no living creature that has these variants because they are fatal. And that certainly there is some, at least some of those exist, and there's some truth in that statement. But put it differently, when it gets to this issue of adopting a structure that does the same thing in the same way, then most likely many variants will have an effect. Put differently, in the extreme way in which I formulated, that you have a variant that is neutral is very unlikely. Or map it back to the problem that we have here. What this shows is 67 in 100 can change. But exactly as you said, Michael, this is very dependent on where in the protein you are and which change you do against which change. And that is not on the level of most variants are somehow neutral. Not true at all. To realize this is realizing the unlikely situation. So evolution, what we see here, is not a random record at all. What we see is what has survived. What has survived is exactly the exceptional cases where the neutral variant happened. That's why we see it. Okay? We still don't quite know what fraction we really have. You may not be that far off. So the reality may be sort of pretty much in the middle between these two paths. But there is evidence that in fact neutral variants are unlikely, and picking 5 in a protein is very, very unlikely. So we can sort of exercise this. Whenever people try this without intelligence, they fail. When people are structural biologists and they think about which residue to change, they can do it. And this is sort of exactly both point in two different directions. One seems to suggest, since if they pick at random, it fails, that most changes have an effect. Since they can change, it points to what you said before, that most variants are neutral. So another way of looking at this riddle is for the sort of proteins that are studied very widely, for which people change really every single position, in every position there is a change that knocks it out. And every position you can find a substitution that is neutral. So somehow both extremes are valid. Okay? Nevertheless, getting back to what this means here. This means that the particular pattern, where can I change which are those 67 residues? What can I change to what? That is relevant information for that particular protein family. And that is exactly the kind of information that we put into our W window. The way we do that is we do exactly the same as before. Before we had this sparse coding or one-hot encoding, where we had 20 amino acids plus one spacer, 21 units coding for every residue. What I show as input here is 13 consecutive residues. And we predict the secondary structure for the center P here. And we replace this one-hot encoding, so-called one-hot encoding or sparse encoding, by the profile. So rather than saying at the center here I have a P, therefore all of these 20 are 0, and P, the 21st unit P, is 1, I will simply look at the P here, look at the alignment, and see how often does P occur in the alignment. In this particular case, actually, it's the same as before. P is 100% conserved, so in this particular case, I will just retrieve the same old hot encoding. But for instance, for this case here, for the E, you see that the actual observed E will only carry 40% of the signal of that family at that position. And in some other cases, like this one here, it's even smaller, so 20%. Anyway, so I use completely the same framework, simply replacing the type of encoding. I simply put the profile into the system. And that is what people typically call the third generation method. In order to use this third generation method, so you begin with the sequence, you have to align, you have to find similar sequences, you do a family alignment, and then you put this into the system here. There is another thing that you can do. So again, there are these two levels. So the first, that input is amino acid sequence, output secondary structure, and the second level, input is the predicted secondary structure, and output the other secondary structure to make the prediction of segments longer. Now, all of this is local information. So what I mean by local is I take out this W window and I accumulate all the information that I can in those W, right? I'm not talking about anything that happens outside of the W. So I'm not looking at what is happening here or what is happening here. That I call global information. And I put this also in as input here. Part of the global information is I take the amino acid composition of the entire protein. Or I look at the length of the protein, it's also global. Or I look at the distance of this W position here with respect to the begin and end of the protein. So this is global information. So the first two units here are such that the global information, wherever this W is the same. The second two, my position of this window here is obviously changing when I move the window through the protein, right? But all of these are global information in the sense that the data I use is sort of collected from something outside, beyond that local window W. Now, I'm sort of making this distinction because there's a surprising finding. And the surprising finding is, okay, Michael Levitt published a paper in '76 that gave him the Nobel Prize, but he also published a paper in '76 in which he simply classified proteins according to the content of helix and strand, the percentage of helix residues and the percentage of strand residues. And I cannot remember whether he did it this way or the other way around. And he looked at he looked at some thresholds, and he said there are some proteins here with a high content of helix and a low content of beta strand, and then there are some proteins with a high content of E and the low of alpha, and he called them all beta, all alpha, and everything in between here alpha beta. Okay? Let's just indicate that by some sort of cloud. Now, we could call that the class, the secondary class of the protein. And in this particular case, we have three classes. So we have a three-state class prediction, all alpha, all beta, alpha, beta. Now we could train a machine learning device to predict the class. Or we could use this device here, predict how many helix residues we have, how many beta strands residues we have, and simply map it onto the figure that Levitt published, right? This is another prediction method. Without changing anything. So now we can assess how well does the global information help me in two different ways. So I assess it in something that is local. Q3 is a local information. Q3 cuts out all the residues, simply asks what fraction of all the residues are correctly predicted in the three states HEL. Yeah, I'm sorry, it's not in the slides at all. It's something that I'm making up while I'm going. In the three-state system, you completely remove the correlation between adjacent residues, right? It's totally local. And then I have a situation where I use global input information and where I do not use it. Okay? And I get a Q3 for this system here and these numbers are made up. But something like that. I get a Q3 of 65 and 65. Conclusion is, same method, global information didn't help. Right? Anybody objects to that statement? In fact, I went one further. I concluded that obviously these two methods are the same. It's not only that it didn't help, but they actually produced the same result. And that turned out to be not true at all. So what I then looked at is this C3. So Q3 is a per residue score. The C3, I call it C3 now, is a classification on the level of per protein. So that classifies what fraction of the proteins are correctly predicted as all alpha or beta, alpha beta. Okay? Are you with me on that? Yeah? And what I saw was 65 and 60. So I saw that when I look at a global measure, entire protein classification is a global measure, in fact, global input information helped. This is remarkable. But it means that something, whenever you do a machine learning device, and you look at some very different type of information as input, global versus local in this particular case, and you ask yourself, does it help or not? And you assess it on something that is local in this particular case, then that may be the wrong way of seeing how much it improves. So the improvement was there. This was a different method. We just did not see it because we didn't look at it in the right measurement, in the right score, in the right matrix. And this will often be the case. It will always be, not always, it will typically be much more complicated for you to find out, is there another way in which I can look at it? But just take this as an experience and think about it. Is there another way of scoring it? Is there another way of seeing that, or finding out whether or not global information or something that you add in terms of information helped you at all? Okay, so it did help in this particular case in a variety of ways. And then there was this issue that we had balanced training led to a Q3 of 60%. Unbalanced, that was before evolutionary information, led to 62%. And that is simply because the unbalanced training did better on the class other, of which there was more in the database. And that's why the Q3 overall was better, but this one was much better on beta. And somehow, it's unclear to use this or with this, which is better. In the end, what we did is we simply averaged over them. Now, the average over different prediction methods is always going to be better than any of these methods, as long as these prediction methods have an uncorrelated white noise. So what that means is that every prediction method makes two types of mistakes: systematic mistakes and what people sort of call white noise, which is sort of random mistakes. Okay? And random mistakes have to do with the way you do the training, with the way you initialize the connections, and all kinds of issues. What type of input features you use. Systematic mistakes could be the type of input features. So if your input space is too small, you systematically will not be able to predict it. And it's a systematic mistake. No matter how many methods you average over, this major thing that you miss. So if you have W only 1, you will not see long-term correlation. And there's just no way, by any averaging over hundreds or millions of these methods, you will still not see it, right? But when you look at the random mistake, the white noise, and if you look at two methods that have a different white noise or are not correlated, so they're not making exactly the same white noise, then averaging over those will get rid exactly of the white noise. Because where they will be agreeing is, in fact, where you're more likely to be right. That's why averaging, you can strictly show that averaging of uncorrelated methods, as long as there's a white noise component, will always improve. So the later tools use more. Michael is going to talk about even more recent tools. So that is the first level sequence to structure, the second structure to structure, and then there is a sort of third level that uses the input of the structure to structure for secondary structure prediction and solvent accessibility prediction, puts all of that together to predict secondary structure. Now, the first level, the first method that used evolutionary information it was sort of the third generation method shown here. It was called PhD then. This is a sort of first generation method for this particular protein SH3. This is a second generation method. The first generation typically is 50%, so this performance is above average. The second generation was 60%, this is above average. This is 72, so the question now becomes, is that above average? The way you typically do that is you take the entire database and you compute the per residue performance. You have, say, 700 proteins, and you simply count for all the 700 proteins how many residues are correctly predicted. That deprives you of seeing the distribution. So you should always find a way to somehow, if you can, plot a distribution such as this one, where this is the per residue accuracy per protein. And this is the number of proteins. In this particular case, you see that 72, the one I showed you there, roughly is the average, in fact the numerical average 72.3 for this particular distribution. So 72, that was an average prediction. But you also see something else. Something more scary. What is it? what am I talking about, yeah?
Maybe that is more or less normal distributed?
Yes, and why do you find that scary? You want something, ideally, so do not assume that if you predict, so maybe you could argue my best distribution would be this, where this point is as far to the right as possible. You will somehow get a distribution. So you will have some that are worse. You will not only get the good ones. So in some sense you will see a distribution, right? And there is some bounds. So the distribution cannot be more than 100. So it cannot be less than 0. Most likely it cannot be less than random somehow for at least the entire distribution, because then you turn the method around. So we somehow will see something that is sort of normal-like, not completely normal. So with that is, yeah?
Yeah, but there is still some outliers.
That is the part that I meant.
Yeah.
So this one, for instance, here, and I cannot remember whether that is the cold shock protein or... So this is the most extreme case here of a protein that is predicted at level 33 or something like that is random. This is clearly below. So there are a bunch of proteins here, at least 2 or 3 proteins that are predicted at levels clearly below random. And I said your prediction can never be less than random, but for particular proteins it can. Because the prediction method cannot be worse than random, because then you flip the labels. But you cannot flip the labels for that random protein, because you would have to flip the labels for all the proteins, and then essentially you would move the prediction, it would make everything worse, right? So yes, you would do it better for this protein, but everything else would become the outlier. So that is completely counterproductive. Now, so as a developer, you will tell the user your average is going to be 72, but it could be that one. Is there anything else you can do to tell the person, well, actually, maybe your protein is that? Or can you give any additional information to the user?
Maybe those proteins have something in common. I'd have to look at them.
That's a great idea. This is what people do. In fact, this was also one of the missions from the beginning in CASP. Once you make a mistake, find out why you did it. But the trouble is, once you know what the right solution is, you very often can explain why you made a mistake. But then changing your method such that the mistake doesn't happen the next time is an altogether different issue. Because then you will often see that if you sort of tweak your method to make that one predicted right, maybe something else becomes the outlier. So this is a very tricky thing. You are totally right. The first thing that you should do whenever you see an outlier, see whether you can explain it. Is there something that is common between these two that none of the others has? So pick the worst 3, pick the best 3, and when you analyze, is there something in common, look at both of those and see whether what you have in the worst 3 is really different from what you have in the top 3. Or ideally different from anywhere else, but clearly it should be different from the top 3, for instance. And that you can easily do. When you look at a single protein, you can look at 3. So there you should start. And by the way, you should always, whenever you have an outlier, try to come up with an explanation why that is such an outlier. In many cases, this is a way to find a bug. Outliers are always the best way for you to understand your results in your data. Always do that. So whatever you do, follow Kira's advice. This is always the first step. But I meant something else here. So say that did not lead anywhere. You're totally right. This is the way you should start. But say that leads nowhere. And by the way, you also have to sort of give yourself a very limited time. Looking at this sort of thing can easily take days. Take a day for it to look at outliers, but take or 2 hours, or find a particular framework of time frame and use it up entirely with doing that, and don't use more. But again, what else could we do? And I shortened my slides, and in shortening I realized now I sort of threw important things away. So I would have to use the whiteboard again. What else could we do? when I look at my prediction, say I have residue I and HEL, and the prediction were something like 0.4, 0.3, 0.3, and I have begun to call that not loop but other. For residue J, I have the example 0.8, 0.1, 0.1. Both samples are predicted helical. Ri and Rj are both predicted to be in helix. Because in both cases, the value, the output unit with helix is the highest value. But the second example, Rj, obviously is much stronger. Right? How could you use that? Yeah?
Maybe if you take all the amino acids and average the probability,
Yes.
and then you can like base approach.
And here we are. That's exactly what this one does. And you see, so it doesn't work quite. So you don't have a clear correlation. This is the performance per protein. This is the reliability index. With the higher numbers mean that the probability is higher. So higher numbers are, this is higher, this is lower. So essentially it's looking at the difference between these two numbers, shown here. And so the worst outliers, the 4 worst outliers, at least 5 or 6 worst outliers are all below some sort of half here, right? The absolute worst are below whatever number here. So there is some correlation. It's not ideal, but clearly you sort of can see that the bad cases are more on that side and good cases are more on that side. That helps you to, if you have a prediction for one particular protein, see whether you're more likely on that side or on that side. That you can do. It's not strictly working. But it's strictly working on a level that is slightly better when you look at per residue ways of plotting it. What I show, what the important part here is this line here. This is percentage of residues predicted, overall accuracy, the Q3. Now, the method, I didn't say that. Yes, I did say that. The overall performance, if I look at all residues, is 72%. That's where we start from. Okay? And now I'm going to look at the half of the residues that are most strongly predicted. So the half of the residues with the highest reliability index. So this is half here. And I see that for that half, I get sort of 86%. Okay? So there you see, this is a very clear line. Again, it's not always a clear line, but to do this line, to look at this analysis, is a cumulative value. Cumulative in the sense, I'm not asking for one particular threshold of this reliability index that I show here, what is the performance, but I'm saying if the reliability index for all those that are higher than this point, what is the performance? Okay? Anyway, it clearly allows users to sort of focus on the most likely correct predictions. And it also allows users to look at the worst predictions. So what, in terms of secondary structure prediction, what is a really bad prediction? I mean, low number, but in particular bad would be if you confuse alpha helices and beta strands. And what this shows here is that only something like 20% of all the proteins have no bad. Most of them have somehow bad predictions. So it always happens. And finding them out is something that you can do sort of guided by this reliability index. Finding out which ones of these bad predictions are likely to be right or wrong. Okay, another thing that you can use secondary structure predictions for, in fact, that is surprising, is this example here from initially Kim, a protein called chameleon. In chameleon, what they did is they took this particular protein here, and in some region here that forms an alpha helix, they changed the sequence into something slightly different, and the slightly different thing then was the same here and there. So they had a stretch of 11 residues that were inserted in two different proteins, two different parts of the protein, and they observed that in two different parts of the protein, one adopted in a beta strand and one adopted an alpha helix. Now, if you use evolutionary information, then you can actually predict this. So you can predict that in this conformation, the same peptide of 11, although the input window is only 13 residues, just a little bit more than this 11, it still predicts correctly that there's a helix here, and it still predicts correctly that the beta strand is in the other position. But it does not do that if you use only amino acid sequences input. Okay. There are now some details here, and I'm going to rush through them, but I'm just going to have some background issues more. I'm going to focus a little bit more on the background issues. So one is if you have a certain level of performance that is quoted. So far I spoke about 72. Later the method was pushed a little bit higher, and Michael is going to talk about even higher numbers that we reach today. But they are all essentially still third generation methods that use much more data. We give some Q3 equals 76. That's an average over distribution. Again, that is the distribution underneath. And it means that this distribution comes with a sigma, which essentially is the width of that distribution, right? And then that leads to the standard error. We know that stronger predictions are more accurate. That's what I showed you already. And there is, however, a big issue. The reliability index is shaped by the input. And the input is shaped by whatever the system learned. It is almost a factor of two too large for single sequences. So if you don't have an alignment, then the reliability index works less well, because it sort of assumes that most proteins, essentially everything it has been trained on, had a multiple sequence alignment underneath. When you now apply it to proteins without multiple sequence alignments, why do you believe it sort of has a high reliability index for single sequences? Or a wrong reliability index for single sequences? Why does the strength of a prediction not work if I, rather than putting an alignment in, I present a single sequence? So this thing has always seen alignments. Why is it stumbling over single sequences, do you believe? Yeah?
Well, maybe it relies too much on these alignments.
There's a great explanation. In fact, this to some extent is true, but there is one on top of that. You're totally right. It relies so much on the alignment that it actually interprets it to be an alignment. And interpret it to be an alignment, if you give a single sequence, means completely conserved. So the system simply assumes that what you have is a family where you have a strong conservation. Strong conservation means strong prediction. But misleading, because there is no conservation whatsoever in this particular case, right? This is the reason behind that one. So the other issue is that if you use single sequences, and that ultimately gets to the same point here, since it's trained on alignment, single sequences are just not as informative. And again, on Thursday, Michael will talk more about that. So that brings us to this issue: what is the best alignment? What's the limit of prediction accuracy? And these 2 points I want to address. So what is the best alignment? How can we figure out what is the best alignment? Yeah?
Maybe it's just sequence alignments. Maybe we should use an error score and just see, is it like a gap score or something, and then see how good the sequences align. So essentially you're asking, or you're thinking about looking at a score that reflects on the value of the alignment for a family, for an entire family. Is that what you're saying? Yes.
That is true, and people are doing that. I don't believe so far we have been able to completely relate, as you describe it, this to secondary structure prediction, because that has to do with what is coming next. Let me first go through the slides and then sort of explain part of the answer to that. So again, what we use here in the evolutionary information is the divergence, is the fact that some residues change. Some residues change, again in this example that I gave with single sequences, means that in the family you have to have change. And if things are conserved between two proteins that have 99% sequence identity, this is trivial. There's no information contained in that, right? So most of these 67, in order to find them, you need sequences that are diverged to the level of really 67 in 100 changed. And that means you want diversity. So one simple answer to what my previous question was, try a bunch of methods and see how well you do. And so these are different alignment strategies, and you see that this essentially is the value of improvement over a single sequence, not using alignment at all. And you see that sort of a simple cluster or a profile cluster, so this is the advanced cluster, is doing much better, and whatever. So at the time people assumed were better methods do better. The database here is Swissprot. If I sort of replace the database of Swissprot by, actually again, so the database is still being Swissprot here, but I may go out to find more remotely similar ones. I could sort of look at BLAS e-values of 10 minus 3, cut it there. Or I could say I accept everything to an e-value of 1. Which means that there are many, many mistakes, many things in this family that will not have a similar structure. So, and you see that still, for most of these cases, this is a higher, or for many of these cases, this is a higher value. So even if you make many mistakes in your alignment, as long as you bring in more diversity, because the families here are larger than those, more diversity helps you. Even accepting clearly mistakes, okay? Another thing that we can look at is, are there any cases where the Q with a smaller family or a smaller cutoff is better? So below the single sequence is better. And there are even some cases in this one here. There are some cases in this one and there are different cases. So both of those benefit in different things. Now comes the alignment against a larger database. And you see you improve even more. Conclusion from all of that: you want the biggest possible database, you want to have as much divergence in your sequences as you can, and you want to have alignment methods that are as reliable as possible, not necessarily of doing the alignment, but of finding many things and somehow doing it right. And divergence may play more of a role than making mistakes. This is unique to secondary structure prediction. For instance, for solvent accessibility prediction, that already is different. It's just, that is what that said for secondary structure prediction that is different, and I leave it there. Still, at the end of the day, where I know that PSI-BLAST is best, sometimes the pairwise BLAST does better. Below the line here, pairwise BLAST does better than PSI-BLAST. Unfortunately, we cannot, for an individual case, say, take this or that. Since on average, this one is better, this is what you do take. We felt it is not easy enough to measure the information content of an alignment, or the simple relation that you had counting the quality of an alignment did not correlate with a performance good enough to hope that we can invest into that to get a good measure and then use that to switch the alignment depending on the prediction. But ultimately, the reason is that divergence helps you so much that maybe the reliability of the alignment may not be so important. Divergence is what you need, and you accept errors. Here again, we have a number of proteins aligned, and this is the most important feature that people use today as number of effective proteins aligned. Effective means that you're not just counting the number of so you have your query, you have some, it's not a circle, you have some construct around it, and that is filled with proteins that are similar to Q. That creates your profile. And effective means you're not counting all the points here, the number of sequences aligned, but you're sort of clustering them. You're clustering all of these and you're counting the clusters. So not asking how many proteins do you have aligned, but how many sort of subfamilies do you have aligned. That is called the effective sequence number. That is shown here. And the higher the number of effective sequence, the higher the performance increase. So the larger the families go, the better the performance. So this led me to, in 1996, I believe, to sort of assume that the limit of prediction accuracy would be something in the ballpark or that we could clearly reach 86%. And this is, in fact, again, Michael will talk about it, that's the level we have reached today. So I believe we can even go a little bit higher than that. But let's get back to this one here. How to assess secondary structure prediction methods. So I believe we already talked about this issue of we need different scores. Standard of truth is DSSP. Now, assess the data set size. So, let's say we have 500 proteins. We get a distribution, and ultimately, the question is if 500 proteins work. So 500 proteins are sort of good enough to estimate performance of secondary structure prediction, roughly. Does that mean that any 500 proteins would work?